We change plant species here, and also we also change continent. I'm going to speak about South America now. The idea now is to present an overview of what has been done in South Africa and Chile throughout the TROPICSAFE project. And here you can see how in South Africa the phytoplasm present in the country, Candidatus phytoplasma asteris, belonging to the 16.1b. These are the typical symptoms on the white grapevine, yellowing of the leaf. Here in the red grape, it also is turning, but the leaf is a bit more red. But it happens not only in the lower leaves, but also in the upper leaves, with symptoms associated with the presence of the viruses of the unrolling of the grapevine. But also in terms of production, we have a dehydration of the resins. These are symptoms present in all the areas in the world cultivating or growing grapevine. There is not much difference in terms of symptoms, but it's important to be able to have a laboratory allowing us to analyze and establish which is the phytoplasm species associated to these symptoms in the grapevine. We can observe these symptoms at the end of the summer and the beginning of the autumn. In South Africa, we have worked mainly on capturing insects with these traps, these yellow traps, sticky traps, and the main insects for the transmission of the Asteris phytoplasm is Mgenia fuscovaria, also associated with a high abundance compared to the rest. We also found an Coronella prolixa, another same one with low abundance, but it could also be associated to the transmission of the same phytoplasm. This is the situation. We have studied also the Mgenia fuscovaria, and we've seen that in the period between 2009 and 2016, it shows three peaks, three moments of abundance along the year. We have three peaks, so to say, of presence of this insect, of abundance, relative abundance. But in the same study, we continued from 2017 to 2019. This is the red line, but due to the drought, the insect has not appeared. We have not been able to find it. In South Africa, in the end, we have defined that Mgenia fuscovaria is the vector of the AY, and Coronella prolixa is a potential vector of the AY. And we have also found it in the herbs Chiloconidactylo, Chinorum dactylum, Proacea, and these insects have been analyzed and found positive in AY. It's important also to do the test of transmission in order to see that this insect is able to transmit AY. This is what we're doing right now to see if this potential vector can be a vector. The situation in Chile is a bit different. We here have the situation. We have mainly 6 or 7 phytoplasms, but the most abundant, the most important one that we found more frequently, higher than the other, is 16 SR3J. This is explained by the study that we did afterwards on epidemiology. We've seen that in some grapevines, the presence of phytoplasm in the grapevine appeared in the vid, but also appeared in other herbs. And there is also a high abundance of Polygonum aviculare on Convolvulus arvensis. We've seen that all these gave a positive result in the same phytoplasm, but also we had to study transmission. We also see in the same grapevines other vegetal species. This Rubus umifolius also positive, Malva, Brassica, Rosa. We see grapevine positive, weed associated to the grapevine positive, and also other in other vectors. The transmission test, showed that Palatanius tresmitosus is vector of the 3J. And there's another insect also present in the grapevine, but less abundance. It's called Bergalia valbina. It's also vector of the 3J. But in the grape vineyards, grape vineyards, we found different species, but these are the most abundant after the ones we studied before, and they were also positive in analysis, this PCR analysis on 3J. And here we started testing transmission. We collected samples. They're always present mainly in the weed, normally associated to the vineyards. But when they're close to the vineyards, they disappear because they eliminate them with herbicides. And this is here the only green plant present, even though it's not the main plant. But we did this in Valparaíso, especially in Casablanca, which is a very important valley in terms of epidemiology. In order to optimize time, due to the big quantity of insects that we found, we tried to make in vitro transmission tests. It was not good because the insects were properly fed. And here are the results. We tested transmission for the 5 insects, both in vineyards and vinca, and we used a high amount of plants in vitro, and then when we did that on the ground, many of these plants died. That's normal. But here we find a high amount of plants that have been previously used for the transmission test. And in all cases, we've seen transmission for all these pathogens of the phytoplasm 3J. So I would say that now we have enough results in order to do a third publication regarding this topic. Symptoms on the grapevine after 3-6 months are mainly deformations in the growing of the leaves. In vinca, the symptoms are very clear after 6 months. And here we have this yellowing, small size of the leaves, etc. So as a conclusion of this advance of the project TROPICSAFE, we see that in Migenia fuscovaria is a vector of AY in South Africa, that's clear, and Acronolera prolixa is a potential vector of AY, and there are studies, ongoing studies to prove whether it's a vector or not. With the use of in vitro assays, we've seen that the assays of in vitro transmission in vitro allow a reduced and more controlled space. That's important. The insects are less stressed. For example, in the previous tests, we used different Ambicephalus cutrus. When we did this test, we used Ambicephalus cucus, but these died in a few hours. But in vitro, this species of insect lives up to 7 days, which gave us the possibility of establishing that it is a vector. Before we thought it was not a vector, but now we do. We have defined that it is a vector due to the last test we did. We can make tests all year round, so if we have insects collected, the most abundant in winter, for example, associated to certain crop, with the in vitro plant, we can do all these tests during the winter or in any other season of the year. So this allows us to make many more tests around the year. It is necessary to have a space for the production of in vitro plants. We keep assessing the assays that we have initiated. In the next months, we will finish the first tests, and the numbers that you've seen before will vary a bit. But we can say that these new 5 species of Cicadelius add up to the Amplificulus as vectors of phytoplasm of the 16SR3J in Chile. And it is present in many plant species, and this situation, due to the epidemiological studies, we found all these species of insects, but it doesn't explain why the 3J is the most prominent phytoplasm in Chile. But thank you very much for your attention. Thanks to Xunta for the support, to TROPICSAFE, and all the colleagues.